Hi there, good afternoon. It's week number 11 and lesson 46 of Lockdown Bioinformatics. Welcome back. Live streams after a weekend, everybody else, whenever it is, where you are. Yes, welcome back. It's good to be here. So, it's a new week and we're going to take a look at some more bioinformatics tools this week, some things that we haven't explored so far. Beginning today with an extremely used set of tools called SAMTools. So when we ran our alignment last week, so last week revolved around running Bowtie 2 to generate a short read alignment to a reference genome. So we downloaded some E. coli sequencing data and we aligned those reads to the E. coli genome using Bowtie 2. And that resulted in a SAM file, which, if you recall, is quite big. Takes up quite a lot of space on the disk, especially compared to the reads themselves, which are subject to gzip compression. Which at the moment our SAM file isn't. So our SAM file is just is plain text. And so consequently, because as we saw last time out when we looked in a little bit of detail at the SAM specification, that SAM file contains all of the data that's in the FASTQ as well as some other data besides. So because it's at the moment it's text on the disk, it occupies quite a lot of space. So what we're going to look at today, using this suite of software tools called SAMTools, is to explore how we work with SAM files, particularly how we compress them, but not only that, not just how we compress them, but also how we work with them more generally. So that's my goals for today, is to take a look at and introduce you to SAMTools, and then use SAMTools to manipulate, particularly to compress our SAM file that we produced last week, but also to do some other things with it as well. So let's make a start. No point delaying, is there? So I've got my VM here now. So I've not restarted this since we did the alignments. We're still sat here in the same login essentially as we were then. And you can see from the ls that we will have run in one of the most recent lessons, we can see the size of the SAM file there. So it's 1.4 gigs on the disk. When you compare that to the size of the FASTQ input, so 189 meg for the first impair, 215 meg for the second impair, clearly the SAM file at the moment is considerably larger than even that combined input. That is because the FASTQ files have been compressed, so we've compressed that text FASTQ data with gzip compression. At the moment, the SAM file is just plain text, and so we could just compress the SAM file in the same, because it's plain text. We could compress it in the same way as the FASTQ is compressed using gzip compression, and that would make it smaller. But there are things about converting, instead of doing that, converting our SAM file to a BAM file, which are advantageous, particularly the ability to index that BAM file and therefore explore it much more rapidly. So that's why we're going to look at SAMTools today. It's the primary reason why we're going to look at SAMTools. It's also an extremely useful piece of software for an awful lot of other things besides. So to begin with, let's let's install SAMTools. That's the first thing that we need to do. So we haven't used it yet, so we don't have, at the moment, we don't have access to it because we haven't installed it. So we're going to install it using Conda, the same as we've been installing all of our bioinformatics software as we've gone along. Clear my screen and run a Conda install. So I'm installing this in my alignment environment, just as we've installed Bowtie 2 in this environment as well, because this is where we've been running all of our alignment sort of exercises. So it's as good a place as any to install this piece of software. So let's do that first of all. So Conda install, and the name of the software is SAMTools, SAMTools. So if you remember, SAM, so SAM stands for Sequence Alignment Map Format. So that's our file format, and SAMTools is a set of software for working with and manipulating SAM files. Okay, so Conda is going to go off and resolve our environment and try and install SAMTools and any dependencies that SAMTools may have. We've seen this quite a few times now, and Conda is behaving reasonably sensibly for us so far, at least for me anyway. Hopefully it's for you as well, and that your software installs are working. I haven't had any significant problem. I suspect that my VM is getting rather full again in terms of disk space, so it might be a good idea to do a Conda clean after this, just to tidy things up a little bit, because I think I'm probably starting to run a little bit short on space in my virtual machine volume. So yes, so there's SAMTools and its dependencies, SAMTools version 1.9, and a few other things besides. So that's going to install, and then once this is installed, we should have a binary on the system, an executable on the system, just called SAMTools. So we'll run that and we'll take a look at the help information that comes with SAMTools. So SAMTools is an executable which works actually a little bit like the thing that we've encountered so far that works most like SAMTools is Conda. So Conda, when we run, in fact, let's run Conda clean because it's a good example of this. So Conda, when you run it, if you just run Conda on its own, it doesn't really do a great deal. So Conda without any extra information will just tell you a list of actions that you can take using Conda. So Conda requires a command to work. So Conda, in this case, we're going to run Conda clean. Okay, so Conda clean, I think it's -- all to clean, just to clean up all of these little packages which we've got sitting around, all of the things that we've downloaded for the packages that we've installed. So that's going to tidy our system up a little bit and retrieve some space. So that is when you run Conda, and then you run a command that you want to run with Conda, sometimes also described as a verb because it's an action that you want Conda to undertake. And SAMTools is the same as this. SAMTools on its own is if you just run SAMTools without any extra information, will, just like Conda just did, will give us a list of the commands that we're able to run with SAMTools. So the way that you run SAMTools is the same as the way that you run Conda. You run SAMTools and then a command, and then all of the options that you want for that you need for that command. So hopefully what you can see here is there's a lot of commands that you can run with SAMTools, so a fair old list of them. And these commands are capable of doing different things, and they're used for different purposes. We're going to explore a few of these commands today, 3 or 4 of them. But there are, as you can see, there are plenty of others that we can explore in other contexts in future sessions. Okay, so at the heart of what we want SAMTools for, right now anyway, is converting from a SAM file to a more compressed representation of the data. So like applying gzip compression to a FASTQ file, we want to be able to reduce the amount of space that our SAM file occupies on the disk. And for these purposes, we're going to convert our SAM file to a BAM file. So BAM stands for binary alignment map, and it is a compressed version, compressed lossless version of SAM. So you don't lose any information in converting to a BAM file. You just make your file smaller effectively. And the tool for doing that conversion is called view. So that's the command that we need for converting from SAM to BAM, and indeed back again. So let's have a look at the help file for view. So SAMTools and then view is the command, and if we run that without any further arguments, it should print out the help, this time not just for SAMTools but for SAMTools view. Okay, so there we go, this is the help for view. So it takes some options which are detailed down below, and then it takes an argument, which is the input file, so in our case a SAM file, in.sam, and then optionally also you can view a region. We'll come back to that later. Okay, so let's have a look at the options. So the most critical one for our purposes this time is that we want our input file is going to be a SAM file. We want to tell SAMTools view that we want our output file to be a BAM file. And for that, our option is -b, -lowercase b. And that tells SAMTools to produce its output as a BAM file. Okay. Then by default, SAMTools will produce its output to standard output in the same way as Bowtie would have done by default. We'll come back to this shortly as well. Something else we'll come back to later on. So by default, will produce its output to standard output, but you can tell it to produce an output file, and you can do that with -o followed by the name of the file that you want it to produce. Okay, so that's the other option that we want. For this particular operation, we don't need to tell it anything else. We just need to tell its output BAM and where to output it to. Okay, so to construct this command, it's SAMTools view. Then we want two options. The first option is to output the results of this command as a BAM file, so -b. Then the second option is to tell it where to put that output, so -o, and that is followed by the name. That -o is followed by the name of the output file. Okay, so the output file, we're going to call it after the SRR accession and .bam because it's going to be a BAM file. Okay, so that's our output file, and then the final thing that we need as part of the SAMTools view command, if we go back up to the usage at the beginning, so we've got our we have our executable SAMTools, our command view, we've added in our options, the -b and -o options, and now we need an input file. So in our case, our input file is our SAM file, and the format of this file is auto-detected. It used to be that you had to tell SAMTools the format of your input file, but for quite some time now that's not been the case. So you used to have to give this a $S option to tell it that your input format was SAM, but you don't need to do that anymore. It's not required. As it says here, it says ignored because the input format is auto-detected. So our input file is our SAM file, which we've already created from our run of Bowtie 2 last week. So that is our SAMTools view command. This will take our SAM file as input. It will produce a BAM file as output because that's what we've told it to do with the -B output, -B option, and it will produce that output into this named file here. Again, because we've told it to do that with that option. So this will run. This will take for a 1.4 gig SAM file on a virtual machine. This will probably take a couple of minutes because this is not it's got a fair amount of data to kind of chew through. So we'll get that running. So yes, reading the SAM file, producing the BAM file on the disk. If we have a look in another terminal, so here we are, we're in the same directory, in our week 10 directory in our share folder, and you can see that the BAM file is there now, and it will be getting gradually larger as SAMTools works its way through the SAM file. So currently it's 52 meg. If I list again, you'll see that it's gone up slightly in size, and it will continue to get bigger as we write the binary BAM output into that file. So that's using SAMTools to convert our SAM file into a BAM file. I should note at this point also about the other file format conversion that SAMTools can do, which is to convert not to BAM but to CRAM. So CRAM is another related format for sequence alignments. CRAM is a little bit more modern than BAM and potentially is better at compression as well, so you can compress your data more with CRAM. Because CRAM is a bit newer, it's not always universally compatible, so it's not always compatible everywhere, whereas BAM kind of tends to be. Everywhere seems to be able to consume a BAM file, whereas that's not universally the case with CRAM. But for long-term storage, especially, some advantages because the compression is better in a lot of cases. So it could be, there are circumstances in which it would be worth exploring the options for storing data as CRAM rather than as BAM. But for our purposes here today, BAM is sufficient. So we're converting to BAM. This is going to take a few minutes just because of the size of our input file. We're up to 430 meg, so actually we should be getting close to being done because that's more than a quarter of the size of the SAM file. So I would expect that that's got to be close to finished, and in fact it has finished. So there you go. So that is that conversion done. And you can see, so okay, so our SAM file is 1.4 gig. Our BAM file is 447 meg, which is much closer in size to the total size of the two FASTQ files that went into making the alignment. So this is obviously much smaller than the SAM file, and it's, as I mentioned earlier on, this compression, so this file format conversion from SAM to BAM, is lossless. So all of the information that was in the SAM file is also in the BAM file. So what that means for our purposes at this point is we can quite safely remove the SAM file. We don't need the SAM file anymore because we've taken a copy of the SAM file effectively into the BAM file. But before I do that removal, I'm just going to check a couple of things, just to make sure that we really have converted correctly. So you can use SAMTools view not just to convert from SAM to BAM, but also to convert back again. And if you've got a BAM file and you just run SAMTools view over it, the default operation for SAMTools view is to produce its output as SAM to standard out. So if we just run SAMTools view and then with a BAM file as input, it will print the SAM, effectively it will print the SAM file to the standard output stream. So there you go, that's what that is doing. And so you can see that this is a SAM file. We looked in detail at a SAM file in the last session in number 45. Because it's producing this without any errors at this stage, I can be reasonably confident that the compression has worked and that this BAM file is a perfectly valid file, because usually if something's gone wrong with writing a BAM file, you won't be able to read it back out again at all. So you will get, when you first try to read the file out, you will get an error message in most cases. So the fact that it can read it at all means that it's almost certainly a valid BAM file. If you really, really want to check, then I'm just going to kill that. Then you absolutely can. You can check. You could check the header. You could check the first few reads. You could check the last few reads. There's. You could write it back out to a SAM file and use a tool called diff. Although that's quite a long and involved thing to do. You could even write it back out and calculate the MD5 sum and compare it to the SAM file. In this case, we're just going to we are going to make the assumption that this is the equivalent of the SAM file, that we've effectively saved a copy, and therefore we can remove the SAM file because that SAM file is very large and I don't want it hanging around for longer than I need to. So I'm going to remove my SAM file at this point. Okay, so that's gone. Now I'm just going to take a little bit of a diversion now and talk about why these tools that I've been talking about very recently, so particularly Bowtie and SAMTools, produce their output to standard out, and how we can use that in nice, meaningful ways. So let's take a look at the command that we ran last week, the Bowtie command that we ran last week. So I'm just going to search my history for my Bowtie command, and I need to grep. There we go. So here we are. This is the Bowtie command that I ran. Number 843 in my history here is the Bowtie 2 command that we ran last week. So let's take a copy of that. And let's write a script that's going to run Bowtie 2. And I'll kind of try and explain why I'm doing this as we go along. So run bowtie.sh. Okay, so I need a hash character. Okay, hold on, I need to find the hash because my keyboard doesn't work to print out a hash, does it? I don't think. No. Right, okay. Bear with me a second. I've got no I've got no script in here, so let's do this. Week 5 must have a script in it. Yeah. There we are. Let's borrow the shebang line from there to paste it in. Somebody asked on Slack, I think, about sharing your clipboard between your host machine and your virtual machine. which is totally possible. And you've already installed, if you've got your shared directory set up and working properly, you've already installed the dependencies for getting shared clipboards working, which is the host tools that we had to install to get that shared directory working. You can then share your clipboard between your host and your VM. So if you go to devices and then shared clipboard and then go to bidirectional, so that your clipboard on your VM is shared with your host and vice versa as well. Clipboard on your host is also shared with your VM. It will require a restart of your virtual machine to make that take effect, which I haven't yet had a chance to do. So I'm not actually sure at the moment whether this sharing is working, because I haven't had a chance to reboot my VM to test it out. But in theory, after your VM's had a reboot, you should then be able to copy things on your host machine and paste them into your VM. Right, so Bowtie 2. So we've got our shebang line. Our Bowtie 2 line looked like that. So let's copy that out as well. And paste it in there. Right, so that was our Bowtie 2 command. The other thing that I have learned how to do since we last wrote a script is how to activate a Conda environment from within a script, and that is you have to source your Conda environment first, which I believe is in I believe it's there. So if you run that, and then you can run Conda activate alignment within your script. I'll just check the path to that. This is just little things which have been asked over time, and I'm just trying to kind of address as we go along. So let's save the output file, check that this file actually exists. Seems to. So that file, that conda.sh file, which is there in your Conda installation, that is the script that is run when you first log in at your terminal. So when you start a new terminal and you log in, that conda.sh script is run to activate your base Conda environment. So within the separate environment of your script, if you want to access a different Conda environment, you have to run that script first. And that's what the source command does in this script. Is it runs that conda.sh script within the environment of this script and provides you access to your Conda environment. So then we can activate our alignment environment should we wish to. So that's how to go about doing that, which is something that we had talked about doing way back when we first started using Conda, but which is something which I've now figured out how to do. Okay, so our Bowtie 2 alignment. I mentioned last week that this alignment, unless you give it the -s, the -s option, will produce its alignment output to standard output. So this saves the alignment to file named in S argument option, sorry, be consistent in our terminology. So that saves the alignment to this named file. But if you don't include that S option, then it would produce the output to standard out. Okay, so so this will produce alignment to standard out. So bowtie 2-x E. coli underscore K12 -1 SRR 11866736 underscore 1 dot fastq dot gz -2 SRR 2z's 11866736 underscore 2 dot fastq dot gz. So that will produce the same alignment. That will produce the alignment, but it will produce it to standard out instead of the file named in the -s argument. So what's the benefit of being able to do that? Why would we want to produce our alignment to standard output? Because in the absence of doing anything else, it would print the alignment to the screen, which would probably exceed the buffer of our terminal, so we would only get the last 1,000 or 10,000 lines of the alignment on the screen by the end. And short of sanity checking that the alignment is actually being produced correctly, how is this useful? And the answer is, it's useful because of output redirection, because we can take that standard output stream, and in the same way as we have done in the past with other tools, we can redirect it into another command. So we can use our pipe symbol to redirect the standard output stream of bowtie 2 into a tool which accepts standard input. And an example of a tool which accepts standard input is SAMTools. So SAMTools, or SAMTools View particularly, will accept standard input. So it will take the input, or it'll take the output stream produced by bowtie and use it as its own input stream. So this is a useful way of never actually having to write that SAM file to the disk. So you saw how big the SAM file was, and this is a relatively small set of reads, 1.4 million pairs. It is, relatively speaking, quite a small amount of data. So this is whole GM sequencing of an E. coli, and so we've got about 300 million bases of sequencing data for a 4.5 million base pair genome. So what's that? That's about 65-fold coverage of the E. coli genome. So if you take that number of bases, divide it by the number of bases in the genome, you get around about 65-fold coverage on average of every base in the E. coli genome. Obviously, the E. coli genome is quite small. If you were to do a 30-fold human sequencing experiment, you're talking about 90 billion bases of coverage rather than 300 million bases. So many fold more, many times more data. And you don't necessarily want to write that alignment out. If you then align that data to the human genome, you don't want necessarily to write that alignment out as a SAM file, because it would consume many gigabytes of disk space. And so you might want to, instead of writing out the SAM file to the disk, you might want to directly convert that SAM file into BAM in memory and only write the BAM file to disk. And that is what this pipe will allow us to do, which is that we can convert the SAM output from Bowtie 2 into a BAM file on the fly. So that is what this command is going to do. 736.bam. The only thing that you need that might be slightly unfamiliar to us, I'm just going to split this across multiple lines so that there we go, just so that we can read it all on the screen. So the only thing that SAMTools needs to be able to consume the standard output stream from Bowtie is a placeholder for where that input stream is going to be consumed. So the way that you do that is just with a single dash. So the dash at the end of this command is saying to SAMTools, put the standard output stream from the previous command here. So this is a placeholder. Instead of putting a file name for a SAM file, this is saying to SAMTools, use standard input, which is the redirected standard output becomes standard input. It says use standard input here. So that you need that trailing dash on this command. And that then will take this, the output from Bowtie 2, and using SAMTools convert it to a BAM file so that we never have to write that big SAM file to disk, which is quite a useful thing to be able to do. Okay, so that's this process, exploiting pipes to be able to not write the SAM file out. Few questions being generated. Size of the SAM output is greater than the memory of the computer, so it should work fine because it streams the SAM a line at a time through memory. It doesn't actually hold the whole SAM file in memory, so it will still write it iteratively to disk, but iteratively as a BAM, not as a SAM. So you shouldn't have to hold memory, so that won't be an issue. Is it still possible to check if Bowtie 2 is successful? So you will still get, at the end of this alignment, you will still get the little alignment report that Bowtie prints, because it prints that to standard error, not to standard output. So that will still be printed to your terminal at the end of this command. So you'll still get Bowtie 2's alignment report. And of course, you've still got the alignment file on disk. So you can still use SAMTools view to look at the contents of your BAM file and check that sanity check that it's done what you would expect. And then the final question, just to tackle that one as well, if the Bowtie command isn't in your history, it will be because probably because you will have shut down your virtual machine without exiting your terminal. So your commands get saved into your history when you, so let me save this file. Your commands get saved into your history when you run exit. So part of the process of exiting a terminal session is to write the command history for that session into your history file so that it's then searchable in future. Okay, so some other SAMTools commands just to kind of keep going with. I said I was going to do 3 or 4 SAMTools commands today, so let's carry on with that. So the next thing that you will typically want to do with a BAM file, once you've converted your SAM file into a BAM file, is you will want to sort it in some sort of order. So at the moment, our SAM file is in the order of the reads that were in the FASTQ. So each read in your FASTQ file is aligned successively to your reference genome. And those reads therefore end up in the SAM file in the order they were in the FASTQ, which is a logical order in one respect for those reads, but actually a sort of more logical and an order which makes more sense in the long term to store things in is in the order that those reads fall on the reference genome, and that's called coordinate sorting. So that's a coordinate sorted BAM file. So coordinate sorting is achieved using the command SAMTools sort. So again, here are the options for SAMTools sort, and you can use sort to sort both in coordinate order, which is the default, or in name order, which is the order which they're sorted already. And there are some tools for which name order is preferable, because if your BAM file is name sorted, then your pairs of reads appear together. Whereas if it's coordinate sorted, that's not guaranteed. So your pairs might get split up because they may align to very different places on your genome. So there are some reasons why sorting in name order can be useful. But coordinate sorting tends to be more useful for a number of reasons, which we'll come to. So SAMTools sort, and then we want some options. We don't actually need very many options. We just want a place to write our final output. So again, by default, SAMTools is going to produce the sorted output to standard out, but we want to be explicit about where it's going to go. So we're going to want to put the output in, let's do SRR 118.66736.sort.bam. And then the final thing, so that's our options. That's the only option that we're going to give it. And then the final thing is the input. So that's our BAM file as it currently is, .bam input file. So this will take our unsorted, or name sorted if you like, BAM file and coordinate sort it. So we'll produce a coordinate sorted version of that file. Okay, so again, this will take a little while, and this is something which can be fairly resource heavy. So you can configure this so that it uses more or less memory, and it will store bits of the sorting in temporary files as it goes, to avoid you using all of the memory on the machine to store data structures as it sorts these in coordinate order. So you can configure that. You can also run sort in a multi-threaded way, so you can give it a number of threads to run on, and that will speed up the sorting. It's not completely linear speed up, but it will go faster if you've got the ability, like we were exploring with Bowtie at the end in the last session, running it on multiple threads. You can do the same with your sorting. You can run sort on multiple threads and it will go faster than just running it on a single core. It looks like actually it's going to be relatively speedy, because it's done the sorting now and it's writing that out to the output file. That's what that little message that's been printed to standard error tells me. So it's just merging the output into the right order and producing me a sorted BAM file in my current working directory. So hopefully this won't take very long to write out, and then we can take a look at the difference between an unsorted and a sorted BAM file. So it's nearly there. promise. Maybe I shouldn't just stop talking now. I know how I'm sure you will love me to fill. So surprised this is taking so long. Nearly there. Come on, little machine. Produce your output. Love how sometimes things go faster than you expect, and sometimes things go slower. There we go. All done. So, that has sorted our BAM file. So now we've got the BAM file stored twice. And if we list our directory, what you can see, we've got our unsorted BAM file, which is here and is 447 meg in size. Our sorted BAM file, despite having the same contents, so it's got the same reads in it, is about 130 megs smaller, so about 25% smaller than our unsorted BAM file, and that's because the sorting of the BAM file enables you to compress it even further. So a sorted BAM file will compress more effectively than an unsorted BAM file. So that's one of the advantages of sorting. Let's just have a look at the difference between these two files. So let's use SAMTools view. First of all, I'm going to look at the headers of the files. So there's an option for SAMTools view, which is -H, which will print only the header of your file. So it won't print the body, it won't print the alignments, it will only print the header. And the header of these two files should in theory be subtly different, but I haven't actually checked this, so let's find out. So there is the header of the unsorted BAM file. So that's the header that we saw last week when we looked at the SAM file, the raw SAM file on its own. If we look at the sorted one, so the big difference here is that the SO has changed from unsorted to coordinate, because this is now a coordinate sorted BAM file. So that's the big difference there. There's no other changes to that header, so it's still aligned to the same reference genome. It's still been produced by the same program because it was Bowtie 2 that produced this alignment, but our header is now telling us that it's sorted. If we take a look at the first 10 reads in the unsorted, so let's pipe our SAMTools view command into head, which will show us the first 10 lines. So these are the first 10 lines of our unsorted file, and you can see that these are from the read IDs, this is the first 5 pairs of reads. So we've got the SRR ID and then .1, .2, .3, .4 and .5, and we have pairs. So we have 1.4 and .4 and .5 and .5. So we have a set that are a set of 10 reads, which is our first 5 pairs. In the sorted BAM file, sorry, the first 10 reads are no longer those 5 pairs, but they are the first. They're the first set of reads, in theory, on our reference genome. But actually, so yes, so this is. You see that this read is mapped to our reference genome at base 1. Okay, so that is the first read because it's mapped to the first base of our reference genome. The second read is not the pair of that first read. So the first read is read number 1020699. The second read is read number 393359. And so this is second in the alignment because it aligns to read to position number 2. So it aligns to the leftmost position of this alignment is base 2 of the reference genome. So the pair of this sequence you can see because it's 2 sequences further along. This is the pair here of this one. So the pairs are not necessarily still together anymore in your alignment because the alignment is now sorted by the order of the bases in the reference genome rather than anything else. So the other advantage of coordinate sorting your alignment is that we can now index this alignment. So we can now produce an index of this BAM file. And we can do that. So first of all, let me get my thoughts in order. I'm going to remove the unsorted BAM file because, again, we've taken a copy of the data. So there is nothing in this sorted BAM file that isn't in... There's nothing in the unsorted BAM file that isn't in the sorted, and vice versa. They are copies of each other, even if the order of the data in them is slightly different. So we don't need to keep the unsorted BAM file. So let's remove that because we don't need that anymore. Okay. But we can now produce an index for our sorted BAM file, and the tool for doing that is, as you might guess, SAMTools index. So SAMTools index will produce what's called a BAI file, and that's an index for the BAM for your BAM file. And an index means that you can look things up in your BAM file in a really efficient way, and I'll show you what I mean by that in a second. So let's produce the index first. So SAMTools index, and then the only really required argument here is the name of your input file. So that's our sorted BAM file. So by default, this will produce an index file which will be called srr11866736.sort.bam.bai, and that file, that little file, will be the index for the BAM file that contains that consists of the same name. Okay, so index doesn't take a huge amount of time, so it should be relatively quick. The additional file that it produces. So here we go, we're indexing our BAM file, producing our BAI file, and that's finished. So ls shows me that as well as the BAM file, I now have this index file, this .bai file. That's usually quite a small file. It's a hell of a lot smaller than the BAM file, so just 17K. But what having the sorted alignment and the index allows me to do is to access portions of the alignment very efficiently. So what I mean by that is I might only be interested in reads that align to a certain bit of my reference genome. So in the case of a human genome, say, I might only be interested in reads which align to one of the chromosomes. Or in the case of this, and I've just got to remember, I apologise for this, I've just got to remember the name of the chromosome to be able to do this efficiently. So let's have a look at the header. There we go. So it's called NC underscore blah, blah, blah. Let's copy that out. So let's say I'm only interested in reads which align to a certain bit of this reference genome. So with an index, with a sorted BAM file and an index, I can now reference regions in my alignment. So by doing chromosome name and then position, so 10, let's say 10,000 to 20,000, this will retrieve all of the reads which align between those two bases. So on my reference chromosome between base 10,000 and base 20,000. So this will subset my alignment to just those reads which align between those two positions. So you can see this last one here aligns at base 19,997. We can count this, so rather than just printing it all out to standard output, we can see that there are about 5,700 reads that align in that region of the reference genome, and we can do that quickly and efficiently because our BAM file is sorted and we have an index for it, which enables us to look up which reads fall in between those positions really, really quickly and really efficiently. And that's another advantage of, rather than just applying sort of standard compression to a SAM file, this is an advantage of storing as BAM and having an index. It allows us to do these nifty things which are kind of dependent on that file format, rather than if it was gzip compressed, in which case all of this stuff would be much more difficult to do. So that's a useful thing to be able to do. Okay, final two things for today, and this is thinking about the bitwise flags that we talked about a little bit last week. So if you recall, in the last session, we did talk about the bitwise flag, which is the second field in the SAM file. So you can see this one here is 147, for instance. And we talked about this Explain SAB Flags website here, which allowed us to decipher those bitwise flags using this box here. You could enter the number into the box. SAMTools actually contains a command which will let you do this deciphering at the command line, and that is SAMTools flags, which will take a bitwise flag and will tell you what values that bitwise flag corresponds to. So value number 147 relates to those particular features of that read. The read is paired, it's mapped in its proper pair, the read is mapped to the reverse strand, and it is flags paired, which will tell you the value of that particular flag. So the integer value of it is 1. This is the binary value of it. If you want to know the individual values of the bitwise of the booleans that make up your bitwise flag, you can also use SAMTools flags to query that as well. And as well as being able to directly query the individual bitwise flags, the other thing that you can use SAMTools to do is to generate summary statistics of each of those flags over your whole SAM file, and you can do that with SAMTools flagstat, which will look at the bitwise flags on all of your reads in your alignment and will generate a set of summary statistics for that alignment. So if we run SAMTools flagstat over an alignment, we will get back a little report which summarizes those bitwise flags across the whole alignment. So this will just take a few seconds to run through that BAM file, and then will produce me back a set of statistics for those bitwise flags. So there you go. How many of them are mapped? So this agrees with the little alignment report that we got from Bowtie 2 when we ran it. How many are properly paired? How many there are in the first in pair? How many in the second in pair? And so on and so on. So that's SAMTools flagstat is also quite useful. So that's all of the features of SAMTools that I wanted to talk about today. So that's that's it for today. That's all I wanted to talk about. So join me again on Wednesday for what happens next, which, as you can tell from my hedging, I haven't quite decided yet. So yes, join me on Wednesday for whatever may come next, and that's it. So keep safe, stay well, and see you all next time. Thanks for stopping by, and